Good morning, students. Today we have studied about inversions. It's a structural changes chromosome, and one type is inversion. Earlier we discussed about duplications and translocations, and now we discuss about inversion. An inversion is produced when there are two breaks in a chromosome and the intercalary segment reunites in reverse order, like the segment rotates at 180 degree. Let us imagine that a chromosome has a sequence 1 2 3 4 5 6 7 8 give rise to another chromosome having order 1 2 7 6 5 4 3 8. The segment 3 4 5 6 7 has been rotated here at 180 degree and giving an inverted order of genes 7 6 5 4 3. A similar hypothetical example using a chromosome A, B, C, D, E, F has been shown in above figure, where due to coiling breaks occur between B and C as well as between D and E, reunion at broken ends may lead to inversion of the segment CD into DC. The inversion can be of two types: pancreatic inversions and pericentric inversions. Paracentric inversions are those inversions where inverted gene segment does not include centromeres. On the other hand, in pericentric inversion, inverted segments includes centromere. In order to remember these terms and their meaning, one should bear in mind that pericentric means surrounding the centromere or on the periphery of centromere. Now, the cytology of inversions. Due to an inverted segment in one of the two homologous chromosomes, the normal kind of pairing is not possible in an inversion heterozygote. In order to enable pairing of homologous segments, a shape of loop is formed by each of the two chromosomes. The kind of configuration will be observed both in paracentric as well as in pericentric inversions. As will be observed, the products of crossing over and the subsequent stages of meiosis will differ in these two kinds of inversions. Paracentric inversion. A single crossing over or an odd number of crossovers in inverted region will result into formation of dicentric chromosome having two centromeres and an acentric chromosome which is without a centromere. Of the remaining two chromatids, one will be normal and the other will carry the inversion. The dicentric chromatid and acentric chromatid will be observed at anaphase first in the form of a bridge and a fragment. Double crossovers and crossovers within and outside inversions will give various kinds of deficiencies and duplications. These will also give rise to a variety of characteristics. Pericentric inversion. In a pericentric inversion, where centromere is present within the inverted segment, The Picatinny configuration observed is similar to the one described above for paracentric inversion. However, the products of crossing over and configurations at subsequent stages of meiosis differ. In this case, two of the four chromatids resulting after meiosis will have deficiencies and duplications. However, unlike paracentric inversion, no dicentric bridge or acentric fragment will be observed. Consequently, at anaphase first, no bridge or fragment will be seen. However, in pericentric inversion, if two breaks are not situated equidistant from the centromere, this will result in a change in shape of the chromosomes. For instance, a metacentric chromosome, which means with centromere in the center, a type of chromosome where centromere is arranged in the center of chromosomal branches, or we can say chromatids at center positions, may become submetacentric and vice versa. Here is the diagrams. In first diagram, chromosome pairing and products of crossing over in a paracentric inversion heterozygote. We will see first at A position and inversion of heterozygotes, then inversion heterozygotes and Picatinny configuration crossing over within inverted segments. Products of crossing over in inverted region. Here in 24.17 diagram, dicentric bridge and an acentric fragment at anaphase 1. And now in this diagram, meiosis in a paracentric inversion heterozygote. On the top is shown inversion loop in an inversion heterozygote. Anaphase first and anaphase second configurations are shown in result diagram A to D, resulting from four different combinations of crossover within and outside the inversion loop. And first, a single crossing over at positions 1 or 2. In B diagram, 4 strands double crossover at position 1 and 2. C. 3 strand double crossovers with 1 crossover at position 1 or 2 within inversion loop, and the other crossover outside the loop at position 3. And in the D, 4 strand triple crossover at position 1, 2, 3. And this is the type of paracentric and pericentric inversion which occurs during the crossing over, and it causes structural changes in the chromosomes in the form of mutations in their offsprings. So, offsprings are different from their progenies due to structural changes in the chromosome which occur due to deficiencies, duplications, translocations, and inversions in the chromosomes. So, it's finished. Thank you, students.